Hello, good evening everyone. Welcome to the week 7 live session of experimental biochemistry. I am Surasree. I am a Prime Minister's Research Fellow working in the Department of Biotechnology, IIT Kharagpur. In today's session, we are going to cover these topics: the basics of protein purification and primary sequence based properties of proteins, how to determine their oligomeric state, and what is actually meant by functional assay of proteins. In the previous class, we were discussing about the several chromatographic techniques and how we can use them to isolate proteins. So now we will discuss like how to isolate the protein, how to begin the protein isolation step with the overexpression of protein. So let's begin. So protein isolation for expressing the protein of interest. For expressing the protein of interest, we use several systems. Like we can use bacterial system, we can use yeast to express our protein of interest, or we can use systems like mammalian cells. So protein, obviously it is understandable that if we express proteins in bacterial systems, it becomes much more easier to handle bacterial cells and to express large quantities of proteins from it. We can also use yeast to express several proteins. We can also use insect cells, but the difference between a bacterial system of overexpression of protein and the insect cell or yeast is that the bacterial cell is a prokaryotic cell. So when we are expressing, or when we are trying to express an eukaryotic protein in a prokaryotic system, we have to take care of certain things like codon bias. So if the codons of the codon system which is followed for eukaryotic system is not exactly the codon system which is followed for prokaryotes as well. So that is why the codon bias has to be taken care of. Then if the protein undergoes post-translational modifications, as we know that post-translational modifications do not occur in bacterial system or in prokaryotes. So therefore, if the protein is not stable without the post-translational modification, then we will not be able to express it in a soluble form in the bacterial cell. So there are certain strategies which are taken. Also, there are certain bacterial strains which are used to optimize codon bias or to express disulfide linkages, so to induce disulfide linkages in proteins, and several of these strategies are followed. So several of these bacterial strains are used to overexpress our protein of interest. Now what do I mean by overexpression? So basically, when we will isolate proteins from this bacterial cell, the bacteria expresses all of its proteins. So the bacteria expresses many proteins together, and together with its own proteins, bacteria will also express our protein of interest. And what do I mean by overexpression? Overexpression essentially means that the protein of interest will be produced in much more quantity in the bacterial cell. And for that, what we use inducers. One very common inducer is IPTG. So what we do is we clone our gene of interest under lac promoter, and then we induce the lac promoter with isopropyl thiogalactoside. So this is called IPTG, and we overexpress our protein of interest. So now for doing that, we have to grow the bacteria in certain culture medium. So what is this culture medium? Usually the most common medium that we use for bacterial culture is LB. This is called LB. It is often known as lysogeny broth, or it is also known as Lurio-Bertini medium. So this LB medium contains these components: tryptone, yeast extract, and NaCl. These are the major components of this LB medium, and the LB medium is prepared in a bottle like this or in conical flasks. And what do we do is we autoclave the medium. Autoclaving is done to sterilize the medium, and then we give a inoculum of the bacteria, and then we allow it to grow. Now for its growth, the factors that influence its growth can be the nutrients, the pH of the medium, which also has to be taken care of, the osmotic conditions, that means the salt concentrations and all, and the temperature. So the usual temperature in which we grow bacterium is 37 degrees centigrade. So we use a machine which is called an incubator, where this incubator is a machine which can actually maintain this 37 degree temperature, and in this temperature we grow our bacteria in this particular LB medium. Now when we now the major components of these medium is tryptone, yeast extract, and sodium chloride. Now there are some macro and micro elements which are essential in this medium, which are like carbon, oxygen, hydrogen, nitrogen, sulfur, phosphorus, sodium, potassium, calcium, magnesium, iron. These are the macro elements. And the micro elements, these are like the manganese, zinc, cobalt, molybdenum, nickel, copper. These are the micro and macro elements which are essential for the growth of the bacterium. Now the temperature, so 37 degree centigrade is the temperature in which we usually grow our bacteria. But when we use these inducers, so or when we try to overexpress our protein, so what we do is we essentially or commonly we lower down the temperature a little bit. So overexpression is done at a lower temperature. Why is it preferred? Because it is not necessary always, but in many cases it is preferred. Why is it preferred? Because in lower temperature the proteins tend to be more soluble. The protein which is being overexpressed, they tend to be more soluble, and because we allow this overexpression for at approximately, like, we can do this overexpression for 4 hours to 6 hours, or we can go till overnight. So when we are suppose incubating with the IPTG, which is an inducer, with that when we are incubating overnight, so we essentially maintain a lower temperature so that the protein remains soluble and the protein is not degraded. So this is how we grow the bacteria in a particular culture medium, and we are using this inducer molecule so as to the protein of our interest is overexpressed in these bacterial cells. Now the bacteria has overexpressed these protein. Now what we need to do is we need to collect these bacterial cells, and then we need to take our protein of interest out of these bacterial cells. Now how do we do that? So the first thing that we do is we lyse our cells, lyse the bacterial cells. So this cell lysis is essential, because if the cell lysis is not done, we will not be able to take out the protein out of the cell. This disruption or cell lysis can be done by several methods, like by increasing temperature, or by changing the osmotic conditions, or by using chemicals. So often a detergent which is called Triton X100, it is used to lyse the bacterial cell. So it's a detergent that we can also use to lyse the bacterial cells. Now there are other techniques, for example, which is called a sonication method. So this is actually a sonicator machine. So there is actually a probe. So you can see this part. This is the probe of the sonicator. Now what does it do? So basically, in a sonicator probe, it generates sound energy in the range of, so it's an ultrasound, so it generates the sound in this range of 20 to 50 kilohertz. And because of this sound energy, so this sound energy actually forms these small bubbles inside the sample. So in this beaker here, we have taken our sample. This sample volume can be small, this sample volume can be less. According to that, we can use smaller probes or bigger probes. Now in this beaker there is our sample, and what we do is we set in this machine, we set particular amplitude, and also we set the time, and we set the pulse. So amplitude can be 30%, 40% or 20%. Time is like for how long we want the sonication to be performed, and pulse is for how many seconds this sonication will actually continue. So pulse can be, for example, like 30 seconds on and 30 seconds off. So what does it mean by on and off? So for 30 seconds this pulse will go, for another 30 seconds it will be off, and then again for another 30 seconds it will be on. So like this in intervals this sonication is actually performed. And one more thing is that since it forms these small bubbles inside the sample, so these small bubbles they implode, and this is a process which is called cavitation. Now because of this, the micromechanical forces it disrupt the cell wall, and it also breaks down the large DNA and RNA molecules. So of course, since this is a sonicator probe which generates sound energy and it breaks down the cell wall, so it also generates a lot of heat. So which is why we do not want our proteins to be to become heated, because it will destabilize the protein structure, or it can cause aggregates of the protein. So to minimize this heat generation, what we usually do is we keep this sample in an ice bucket. So we keep this sample in an ice bucket, so there is less heat generation. And also this is a soundproof chamber. So since it is ultrasound, so it is actually not good for our ears, so that is why we usually put the sonicator probe in this soundproof chamber, and then we allow the sample to be sonicated so that the bacterial cells can be lysed by this sonication method. Now of course, after we lyse the bacterial cell, then all the proteins and all other molecules of the cell, all of them will come out in the solution. Now what we can do is, first we have to check whether our protein has been really overexpressed or not by using the inducer molecule. So for that, to check the overexpression of the protein in this cell lysate, what we do is we run SDS page. In the previous lecture we had discussed about SDS page. If anyone needs any details about it, please go to my YouTube channel, and you can go back to the week 6 live session, so where we had discussed about the principles of SDS page. Now we run the protein lysates in SDS page, and we check whether our protein of interest is expressed in high amounts. So this is a gel image. You can see that this is a SDS page image where this LEN1 is uninduced, which means it is a control sample where no IPTG was given, right? So uninduced means no IPTG or no inducer molecule was given in this particular cell, in this sample. And these two LENs, LEN 2 and 3, these are both induced samples, which means there was like IPTG was added. IPTG, 1 millimolar IPTG was added in these samples, and this induction was carried out at 37 degrees centigrade. So both these induction was carried out in 37 degrees centigrade. Why? Because actually this is a protein of bacteria. So this is actually a bacterial protein which was overexpressed in bacterial cell. Why? Because we need the protein in larger amount to perform functional assays with the protein. So since this is a bacterial protein, we had actually overexpressed this protein or induced this protein formation at 37 degrees centigrade only, not at a lower temperature. But usually when we actually express human proteins, for example, in bacterial system, we usually prefer lower temperature. So it can be from 37, it can be 16 degrees centigrade, 20 degrees centigrade or so. At this lower temperature we use for eukaryotic proteins. It is preferred. It is not always essential, because these eukaryotic proteins can also be very well overexpressed at 37 degrees centigrade, but this is an usual practice. Now you can see that this is our band of interest. This is our protein of interest. Because why? Because in the uninduced sample there is no such band present. So which means this protein is not present in the bacterial cells normally, where there has not been any induction, or the inducer has not been added. So either this protein is present, or it can be either it is absent or it is present in very small amount. So it is not present. We cannot see this in the uninduced LEN, but in the induced LENs we can see this protein of interest. And we can see that this sample was induced for 4 hours and this sample was induced for 6 hours. So we can see in 6 hours the protein amount has been increased, right? So this is our protein of interest, which we check by and we check the overexpression by running an SDS page. And this is actually the molecular weight marker. So this is a molecular weight. So this is a 15 kilodalton. This band is of 15 kilodalton, and this band is of 25 kilodalton. And we know our protein is actually 20 kilodalton in weight. So our protein band has come between 15 to 25. So by analyzing the marker band positions also, we can determine that this is our protein which has been overexpressed. So this we are checking by SDS page. Now the next step involves the isolation of the protein and how we can isolate the protein. So for protein isolation, one important component of this protein isolation process is the buffer. And what are the components of this buffer? Now the pH of the buffer is a big factor, of course, the pH of the buffer. And the proteins actually they have their specific pI values, and we know that in the pI value the protein is when pH is equal to pI, then the protein has both positive and negative charges, so that it exists in a zwitterionic form. So when the pH equals to pI. Now since it exists in a zwitterionic form, it can also be precipitated in native form very close to their pI. So the buffer pH that is essentially maintained for protein isolation, it is not exactly, it should not be very close to the pi value. So because proteins can be precipitated in if it is if the pH of the buffer is very close to its pi value. So we maintain a pH which is not very close to its pi. Now we also know that when the pH is less than pi, then the protein becomes positively charged, and when the pH is greater than pi, then the protein becomes negatively charged. And in the week 5 live session, we were discussing about the chromatographic techniques, and we had talked about the ion exchange chromatography. Now we know that in ion exchange chromatography, the protein is either positively charged or negatively charged, and depending on that we either use anion exchange chromatography or cation exchange chromatography. Now to maintain this anionic form or cationic form of the protein, we also have to maintain the pH of the buffer. So if the pH of the buffer is less than the protein pi, then the protein will be positively charged, and for a positively charged protein we can use a cation exchanger, and for a negatively charged protein we can use the anion exchanger. Now proteins can also be salted out. So salting out of the protein is basically one more thing, that is the pH and the solubility curve of the protein. It looks like this. So the solubility is high, but when it is farther away from the pi, but when it is very close to pi, the solubility becomes very low. So this solubility curve also has to be taken care of when we are preparing buffer for protein isolation. Now in case of salting out, what happens in salting out? So basically, suppose this is our protein molecule. Suppose this is our protein molecule. So our protein molecules are covered by water molecules. So this is actually the hydration shell. This is the hydration cell which actually covers the protein molecules, so the water molecules of the solvent or the buffer. Now when we actually add high concentrations of another ion, then what happens, this ion actually so suppose we add NH4 plus ion, then what happens when we keep on adding high concentrations of this NH4 plus ion or ammonium ion, then the water molecules also begin to form a hydration shell around it. So now the more NH4 plus ions or the more ammonium ions we pour into the solution, into the protein solution, the more of the water molecules will go away from the protein, and it will start covering the ammonium ions. It will start forming the hydration shell around the ammonium ions, and as we increase the concentration of this ammonium ion, then more and more water will be drawn away from the protein sample, from the protein molecule. So when more and more water molecules are drawn away from the protein, then what happens, the hydrophobic patches of the protein, they become exposed. And since this hydrophobic patches, so the non-polar interactions, so since the water molecules are drawn away, the polar interactions reduce of the protein and the non-polar interactions become more prevalent. And when these non-polar interactions become more prevalent, then what happens, it comes closer to other protein molecules, and then these protein molecules can together form an aggregate, or it can precipitate down. So that is called the salting out. So by salting out, what we do is we essentially precipitate a protein. So suppose our protein that we have expressed in the bacterial cells, it is not soluble. So in the bacterial cell it is expressed but it is not soluble. So in that case what we do is we essentially salt out the protein. So we use ions like ammonium. So we use compounds like ammonium sulfate or guanidinium hydrochloride. And why do we use these ions? So that or why do we use this compound? So that these compounds can create the hydrogen shell around them, and then the protein molecules can start precipitating. So this salting out is actually which ions to use. It can be followed by this Hofmeister series. So we can actually predict the stability, the denaturation or the destabilization of the protein can be followed by following this Hofmeister series, and which ions we can use, we can predict from this Hofmeister series. Now next thing is when we solubilize the protein or when we take it out in the precipitate, the chromatographic methods we will now use. Now the mostly used chromatographic method is affinity chromatography. For affinity chromatography, the proteins has to have a tag. So usually this is called a 6x histidine tag. So 6x is nothing but 6 histidine residues in tandem. So consecutive 6 histidine residues are present. So this is a tag which actually comes from the vector or the plasmid. So the plasmid in which we express our gene, or the plasmid in which we have cloned our gene, from that plasmid, this 6x histidine tag or the 6 histidine residues, this tag actually comes, and it becomes it becomes attached to the N terminal or the C terminal of the protein, either at the N terminal or C terminal of the protein. This 6 histidine tag is attached. And why are we using this 6 histidine tag? So in while discussing affinity chromatography, I had talked about this, that the histidine residues, now it can bind to the nickel ion, nickel 2+ ion. Now the resin which is used for affinity chromatography, it is a nickel NTA resin, or the matrix, right? The matrix. So nickel NTA is what? Nickel NTA is nickel nitrilo-triacetic acid. So it is nitrilo-triacetic acid associated with a nickel atom. So this nickel atom can now bind to the histidine residues. Basically there are 6 coordination bonds that this nickel atom can actually form, and with 4 coordination bonds it is associated with the matrix. Now the 2 coordination bonds that are actually not fulfilled, so or that are empty, with these 2 coordination bonds it can attach to the 6x histidine residues on the protein molecules. So the protein now gets attached to the matrix. Now how can the protein be eluted out? By using a buffer containing high concentration of imidazole. So imidazole is a compound which is very similar in structure to the histidine amino acid, because the histidine also contains this imidazole ring. Now imidazole acts as a competitor with the protein molecule which contains this histidine residues. So by passing higher concentrations of imidazole, we can elute out our protein of interest from the matrix. Why? Because the imidazole will now compete with the protein molecule, and eventually it will help in eluting out the protein molecule from the matrix. So that is a very common process which we use in affinity chromatography. And I had also mentioned while discussing chromatography that sometimes we use several chromatographic methods together, which means after one by one. Why do we do that? We do that to increase the purity of the protein sample that we are purifying. So often it is done that affinity chromatography is followed by size exclusion chromatography. So in affinity chromatography, the principle is that the protein molecules which have these affinity tag or the 6 histidine residues tag, they will bind to the matrix and the other proteins will be eluted out. And after that, by using higher concentration of imidazole, we can elute out our protein of interest. Now, with our protein of interest, there can be several other proteins also which can come out with our protein of interest. Why? Because many bacterial proteins also contain these histidine residues, because histidine is a common amino acid. So many other proteins can also contain histidine residues. So these proteins which contain histidine residues, which are bacterial proteins, they will also bind to the matrix, and when we are eluting with higher concentrations of imidazole, they can also come out in with our protein of interest. So they will not be they are not desired for us. So we need to exclude these contaminant proteins. For that, we use another chromatographic separation method after affinity chromatography, which is usually size exclusion chromatography. And in size exclusion chromatography, proteins are separated based on size and shape. So there are these porous beads which are by which we fill the column, and these porous beads have a particular cutoff value. So the proteins which are larger than the cutoff, they come out in the void volume. And then after that, the proteins which are so proteins actually gets into these pores of the matrix, and by passing buffer, we try to elute out our proteins. And now these proteins which are larger, they will elute out first. So the larger proteins come out earlier, and the smaller proteins elute later. So this is how a chromatogram of size exclusion chromatography looks like. So here you can see that it is in x-axis there is volume, which means the retention volume of the protein, which means for how long it is retained in the matrix. And this is an in the y-axis there is absorbance, which is in an arbitrary unit. So this absorbance actually tracks the UV absorbance, the absorbance of at 280 nanometer, because we know that proteins absorb UV light at 280 nanometer. So we track the absorbance at 280 nanometer. There is a UV detector which is associated with this chromatography system, and we can see that at approximately 80 ml of volume, our protein is being eluted out, and it is giving us a sharp peak, and by which we can calculate the retention volume of the protein. And also by that retention volume, we will also be able to calculate the molecular weight of the protein. So by using the standard curve. So for standard curve, what is done, many proteins which are of known molecular weight, they are passed through the column, and their retention volume is plotted, and from that we can actually calculate the molecular weight of our protein of interest from this chromatogram, which is a size exclusion chromatogram. And oftentimes what we see is that this peak is more or less sharp, and this peak does not show, this chromatogram does not show much other contaminants. But oftentimes we see that suppose there is a smaller peak like this before the main peak. So this smaller peak actually will be the contaminant, or it can be a degradation product. No, not a degradation product, but it can also be another oligomeric product. it can be an oligomer of the same protein, or it can be contaminant, or often after the peak of our protein of interest also we can see this another actually peak, which can be a degradation product. So degradation product of the same protein, which means the degradation means what? The protein has degraded, so because of that the protein has now become smaller in molecular weight, and since it is smaller, so it is being diluted after the actual peak. So this is the native form of the protein, suppose. And contaminant means it can be other proteins, or it can be an oligomer of the same protein. So suppose this peak is given by the monomer of the protein, but if the protein also exists in the solution as a dimer, then this dimer has higher molecular weight, so the dimer can come out earlier, like this. So by analyzing the chromatogram, we can actually determine several things related to the protein. We will come again to this oligomer part. Now these chromatographic methods, along with the ion exchange chromatography, we can also use. And so then after this protein is purified, now there are several properties of the protein which we can determine from the primary sequence of the protein. For example, the molecular mass. So approximately per residue molecular mass is considered as 110 Dalton, which is the average molecular mass of one amino acid residue. So from that, suppose our protein has 100 amino acid residues, then what will be the molecular mass? 100 into 110 Dalton. That will give us the molecular mass of the protein, average molecular mass. Now the molar extinction at 280 nanometer wavelength, which is given by this epsilon 280. This can also be determined from the primary sequence. Now why is that so? Because the absorbance at 280 nanometer wavelength is given by these amino acids tyrosine, tryptophan, and also to some extent cysteine. Cysteine is nothing but the two cysteine molecules joined by disulfide linkage. So tyrosine is an amino acid which is an aromatic amino acid, and its molar extinction coefficient is 1490 mole inverse centimeter inverse, and tryptophan has highest molar extinction coefficient, which is 5500 mole inverse centimeter inverse, or 5500 mole inverse centimeter inverse. So because of which tryptophan is the amino acid which is responsible for giving the most absorbance at 280 nanometer wavelength. So if a protein has tryptophan residues, they will absorb UV light at 280 nanometer wavelength, and by which we can calculate the concentration of the protein sample. So in our initial sessions we had discussed about this Lambert-Beer's law, which is absorbance equals to epsilon, which is the molar extinction coefficient, into concentration into path length of the cuvette. So this molar extinction can also be determined from the primary sequence of the protein, and so basically how many tryptophan residues are present or how many tyrosine residues are present. Based on that, so suppose there are 2 tryptophan residues, so what we do is we take the extinction coefficient as 2 into 5500, like this. Or if there are, yes, phenylalanine also absorbed in 280 nanometer UV light, but phenylalanine has a very low absorption molar extinction coefficient, so it gives a very less absorbance compared to tryptophan and tyrosine. So if there are 3 tyrosine residues, for example, we can write it as 3 into 1490, the extinction coefficient we can calculate like that. So the total extinction coefficient of the protein can also be determined from this primary sequence, as well as the pi, because the pi of the protein, because the pi is based on the pK values of the amino acids, and if we know the amino acid sequence, then we can calculate the pi of the total protein also. So these are the information which we can obtain from the primary sequence of the protein. Now there is a server which is called the XPASI. This server, there is another section of the server which is called ProtParam. So ProtParam is a web tool in which, if we provide the sequence, the amino acid sequence like this. So this is an amino acid sequence of a protein, and this is actually a protein of 178 amino acids. So what we have done is that we have provided this amino acid sequence in the ProtParam tool. Now how do we obtain this amino acid sequence? We can obtain this amino acid sequence from NCBI protein server. So from this we can obtain the amino acid sequence. It is available online if the protein sequence is known, and this protein sequence we can provide in this ProtParam tool, and it will give us several information. So what are the different types of information that it can provide? That is, first of all, the number of amino acids, it will give us 178, and also the molecular weight of the protein. So in this case, the average molecular weight is not calculated, it is the accurate molecular weight is calculated. So which means by calculating the molecular weight of each amino acid, by adding the molecular weight of each amino acid. The next important information that we get from this is the theoretical pi. So the pi is calculated based on the pK values of the amino acids, and the theoretical pi comes out as 5.44 for this particular protein. So by putting the amino acid sequence in this way, in this server, we can calculate the pi of all the proteins. Now it also gives us this amino acid composition, so which means it is showing us how many amino acids are present and which amino acids are present more and which are less. So for example, there are 13 alanine in this residue, 10 arginine molecules, 8 asparagine, 8 aspartate, there is no cysteine, then there is 18 glutamine. Like this will give us the composition of the amino acid sequence. Now also it will give us the total number of positively charged residues and total number of negatively charged residues, aspartate and glutamate, and arginine and lysine. Now the other important information that it can give us is also the atomic composition, the how many carbon, how many hydrogen atoms are present, how many sulfur and all that. The total number of atoms is also given. Now the extinction coefficient, the molar extinction coefficient is also given for this protein, which is measured at 280 nanometers. So this is the epsilon 280. So this value here is given as 27960. So that is the molar extinction coefficient of the protein. So if we determine the absorbance value of this protein as 0.4, for example, and then how will I calculate the concentration of the protein? So we have this epsilon 280 as 27960, and then how do we calculate the concentration? So we know, according to the Beer-Lambert law, it is absorbance equals to epsilon into concentration for constant path length. So what we do is we essentially divide the absorbance by the epsilon, which is the extinction coefficient value, and then so it will be like this, 0.4 by 27960 molar. So that will give us the concentration of the protein sample, and it also gives us the information about estimated half-life, and the aliphatic index, or the grand average of hydropathicity, so how much hydropathic this protein is. So these all are the information which we can obtain from this XPASI Prot Param tool. Now after we have purified our protein, the next thing that comes is how do we determine the size of the protein. So there are several methods that we can use for determination of protein size. One is SDS page, which is very common. So we run the protein sample in the SDS page gel, polyacrylamide gel, and we also run a protein marker along with it. So this protein marker contains the known proteins of known molecular weights, and by analyzing the position of our band, the band of our protein of interest, from that we can analyze that how much it is of its molecular weight, how much is the molecular weight of our protein of interest. Now there is another method which is this gel filtration or size exclusion chromatography that I was talking about earlier. That is, by the size exclusion we can know the oligomeric state of the protein and the size of the protein. How do we determine that? So this is again a chromatographic chromatogram. This is a chromatogram of our protein of interest, which is approximately 20 kilodalton in weight. So our protein weight is 20 kilodalton, and we have run this protein in an S200 size exclusion column. So then what we from the graph of retention volume with absorbance, we can see there are two peaks that have formed here. One peak at approximately 80 ml of retention volume and another peak approximately at 60 ml of retention volume. Now from the standard, which is the calibration standard or the calibration curve of this particular column, chromatographic column, from this standard we can determine the molecular weight of these peaks, the molecular weight of the protein in these peaks, by analyzing the retention volume. And to understand this calibration curve in details, please go back to the chromatographic techniques session, which in which I had discussed about it. And from this retention volume standard curve we can determine that this peak is actually it is of molecular weight approximately 80 kilodalton. From the standard curve we can determine this, and this peak has a molecular weight of approximately 120 kilodalton. Now our protein, we know that our protein is of approximately 20 kilodalton. So since our protein is giving a peak at this particular retention volume, so therefore we can state that our protein is existing as a tetramer. So this peak, this longer peak, is actually showing us that the majority of this protein is existing as a tetramer. But along with that, our protein also exists in a hexameric form, which is why it is giving us a peak at approximately a smaller peak at approximately 120 kilodalton of molecular weight. So this is 20 into 4, so that means we are assuming that it is a homotetramer of our protein, and this is 20 into 6, so we are assuming it is a homohexamer of our protein. So by analyzing this kind of a chromatogram, we can determine what is the size of the protein and also what is the oligomeric state of the protein from size exclusion chromatography. Two other techniques we can use, that is dynamic light scattering and sedimentation analysis by analytical ultracentrifuge. So what is dynamic light scattering? Dynamic light scattering helps us to detect the particle size and the homogeneity of the sample. So this dynamic light scattering depends on this Stokes-Einstein equation. So here there is a laser of single frequency which is which gives this light, and the sample is taken here in this tube, and the laser shoots its single frequency light, and then there is a detector. And since this sample, in this sample the protein molecules or whatever molecules are present in this sample, they will always be in Brownian motion. So they will always be in Brownian motion inside this sample, and since their particles are in Brownian motion, and this Brownian motion will also depend on the size of the particles. So the larger particles will have less speed and or less diffusion, and the smaller particles will have more diffusion. So this Brownian motion is dependent on the size of the particle, and because of this Brownian motion, the light which is shining upon the sample from the laser, this light will be deflected. Now this deflection of the light can be detected by this detector. The deflection at a certain or the scattering of the light at a certain angle can be detected by this detector, and this scattered light, this intensity of the scattered light is plotted with respect to time, and by which we can actually determine the diffusion coefficient of the sample. So by using this Stokes-Einstein equation, so D stands for diffusion coefficient of the sample, Kb is Boltzmann constant, T is the temperature of the sample, eta is the viscosity, and RH is the hydrodynamic radius. Of the sample. So from this dynamic light scattering experiment, we can determine the diffusion coefficient as well as the hydrodynamic radius of the sample. And from this DLS experiment, we can also determine the homogeneity of the sample, as well as if the protein is existing as a oligomer, that also we can detect by the size of the particle. So from this dynamic light scattering. The next other experiment which we can perform for determining the diffusion of these macromolecules like proteins under centrifugal force is sedimentation analysis by analytical ultracentrifuge, or it is called AUC. So analytical ultracentrifuge is a very high speed centrifuge machine. So approximately its speed varies from 40,000 to 60,000 rpm. That is the highest, the maximum speed, 60,000 rpm revolutions per minute. So what happens here is that suppose this is a sample chamber, and in which we have taken our sample, and suppose here, for example, here is the center of the rotation, and the sample is rotating like this in a so it is rotating like this. So in a this is the speed of rotation, that is omega, rotations in radians per second. Now when this sample is actually under the centrifugal force, there are two other forces which also act on this sample. One is the buoyant force, another is the diffusion force. So and the centrifugal force acts in this way and the buoyant force acts in the opposite way, and this will be the direction of sedimentation. So our samples, they will be sedimented in this direction. So this is the direction of sedimentation. Now from this from this equation we can calculate the sedimentation coefficient of the sample, and we can also calculate the mass of the protein, because this sedimentation coefficient will be dependent on the mass of the protein. So from this analytical ultracentrifuge machine, where the sample is being rotated or centrifuged under very high speed, this sample is being centrifuged, and actually there is live monitoring of this sample inside this machine. So there is a these sample holders, or these sample chambers that we use, they are transparent, so UV light can shine through it, and we can calculate the UV absorbance of this sample. So if it is a protein sample, it will absorb at 280 nanometer, and from this absorbance we can actually determine the sedimentation coefficient of this protein, and we can also determine the mass of the protein molecule by analytical ultracentrifugation. There is another technique which should also be mentioned in this lecture, because this is a technique which is called circular dichroism or CD spectroscopy. This CD spectroscopy comes in handy when we have purified our protein, and when we want to check if the protein contains its native folding or not, whether the protein is denatured or whether the protein is properly folded. To check that, we can use this method called circular dichroism. And what happens in circular dichroism is that we can estimate the percentage of secondary structure, that is the percentage of alpha helix, beta sheets and random coils in our protein of interest. So what is the principle of this circular dichroism? So from the light source, white unpolarized light is passed through this polarizer and monochromator. So monochromator, what it does is it selects a single wavelength of light, and what polarizer does is that it selects a single polarized light, not unpolarized light. Now then this polarized light, this circularly polarized light, will have two components. One is right circularly polarized light, another is left circularly polarized light. The circularly polarized light has two components, and a molecule which is chiral, for a chiral molecule, it will have a preference of absorption of either right circularly polarized light or a left circularly polarized light. So either it will absorb right circularly polarized light more or left circularly polarized light more. So this difference in absorption of the RCP and the LCP can be detected by this detector. So when we pass this right circularly polarized light and left circularly polarized light through this sample, then the difference in absorption, which is actually calculated in millidegrees, this difference in absorption can be detected by this detector. And by analyzing this difference in absorption, by analyzing this millidegree, we can estimate the secondary structure of the protein, because there are in this protein molecule, as we had discussed before when we were discussing protein structure, we had talked about this phi and chi bonds. Now this phi and chi bonds, they have this pi to pi star transition or n to pi star transition, and they also have a preference to absorb left or right circularly polarized light, and so which is why these particular secondary structures like the alpha helix, the beta sheet or the random coil, they actually show differences in or they show peaks in different wavelengths of UV light. So the secondary structure percentage can be determined from this spectroscopy, which is called circular dichroism spectroscopy. And the protein folding can also be taken care of, or the protein folding can also be analyzed. So whether the protein is properly folded or whether it has denatured, that we can determine from circular dichroism. However, we cannot get this residue specific information from circular dichroism, but it is a good method for understanding the secondary structure conformation of the protein. Any doubts so far? Okay. Um, so the last thing that we will discuss today is the functional assay of proteins. So now we have purified our protein. Now we want to perform a functional assay of the protein. So how the protein is functional, for experiment, for analyzing that, we perform these assays. And there can be several types of assays. So if the protein is an enzyme, we can give a particular substrate of the protein and determine how the substrate is being utilized by this enzyme by several techniques. It can be a spectroscopy based technique, it can be other techniques as well. So suppose in our case, in our lab we usually work with ATPases. So ATPases are what? So they can actually, these are actually enzymes which hydrolyze ATP, to perform their specific function. So this ATPase activity, the kinetics of this ATPase activity, or this rate of ATPase activity, we can determine from this functional assay. So where this is a coupled enzyme assay. So what we usually do here is that we have this substrate called PAPE, which is phosphoenol pyruvate. So PAPE is a substrate for pyruvate kinase, for this enzyme pyruvate kinase. So pyruvate kinase converts phosphoenol pyruvate to pyruvate, and while it converts phosphoenol pyruvate to pyruvate, so essentially what it does is that it takes out a phosphate group from the phosphoenol pyruvate and converts it into pyruvate. So this phosphate group, what it does is that it allows this addition of this phosphate group with an ADP molecule, and it converts ADP to form ATP, or adenosine triphosphate. Right now, the protein of our interest, which is an ATPase, this ATPase, what it does is that it hydrolyzes ATP. So it will convert ATP to ADP. Now, if in this assay system we have our protein of interest and we have incubated with phosphoenol pyruvate and as well as pyruvate kinase, then what will happen? This pyruvate kinase will convert phosphoenol pyruvate to pyruvate, and in this process it will generate ATP. So it will allow this formation of ATP by joining the ADP molecule with the inorganic phosphate. So it will form ATP. Now we have in our system our ATPase of interest, which will now convert this ATP to ADP. So the more our protein is present in this assay system, the more ATP will be converted to ADP, and also this pyruvate kinase will also keep on adding, this pyruvate kinase will also keep on converting this ADP to ATP. So then if this assay was still this part, then we could not have detected the activity, the ATPase activity. So to detect the ATPase activity, we have added another, we have coupled another system with it. In this system, what we have given is that this pyruvate is being generated by pyruvate kinase. Now we have added another enzyme in this system, which is LDH, or lactate dehydrogenase. Which is LDH. So what lactate dehydrogenase does is that it converts NADH to NAD plus while it converts pyruvate to lactate. So now since there is our protein of interest in the system and also phosphoenol pyruvate and pyruvate kinase, so what will happen, this pyruvate kinase will convert phosphoenol pyruvate to pyruvate. Meanwhile ATP will be generated. This ATP will be converted again to ADP by the ATPase, and this circle will go on. Now this pyruvate which is being generated by this pyruvate kinase, this pyruvate will be converted to lactate by the help of LDH or lactate dehydrogenase, and in this process this lactate dehydrogenase will actually cause dehydrogenation of NADH to NAD plus. Now NADH gives an absorbance at 340 nanometer. So NADH is the absorbance of NADH can be detected by a spectrophotometer. So here what we do is we use a spectrophotometer and we perform this assay in the cuvette, inside the cuvette of a spectrophotometer. And why, if our protein is being functioning, if our protein is functional, if it is hydrolyzing ATP to ADP, then what will happen, more and more NADH will be converted to NAD plus, because this NAD plus cannot be converted back to NADH. This conversion is not possible. So therefore the more our enzyme is active, the more it is present, the more in concentration it is present, or the more rate of this enzyme, the more will be the reduction in this NADH absorbance. So this NADH absorbance we will track and we will analyze by using the spectrophotometer this NADH absorbance, and we will see that since our if our enzyme is active or if our enzyme is functional, we will see a reduction in the absorbance of NADH molecule. So this is our functional assay, which this is one of the various types of functional assays which we can develop for our proteins, and depending on the action of our protein we can or the function of our protein, we can design our functional assays, and we can determine the activity of the protein, whether it is an ATPase activity, whether it is some other enzymatic activity, we can measure that. So this is a assay system, coupled enzyme assay system, to measure the ATPase kinetics of the ATPase of our interest. So I hope this is clear. Okay. So now we will go on to our question answers. First question. Okay, correct answer. The bacterial growth depends on all of these following factors. Next question. Right, lysogenic broth does not contain agar. Next question. Correct. So a micronutrient for bacterial growth is cobalt. Next question. Right. The correct answer is sound energy is used for sonication process for cell disruption. Okay. The correct answer is pH. When pH is less than pi, the protein will be positively charged. Next question. Right. So in size exclusion chromatography, proteins are separated based on their size and shape. Next question. Okay. So there are three proteins: 50 kilodalton, 30 kilodalton, and 12 kilodalton. So in what is the principle of size exclusion chromatography? Larger proteins will elute out first and followed by smaller proteins. So based on that, the 50 kilodalton protein will be eluted first, then followed by 30 kilodalton and 12 kilodalton. So that is the correct answer. Good. Next question. Right. Imidazole can bind to nickel ion. Next question. What is the answer here? D. So the protein is expressed in bacterial cells and the bacterial cell is lysed. Then to check the overexpression, the best technique to use is the SDS page. If the protein is present in the cell lysate. So we can determine that by running the uninduced and the induced sample together. So in the uninduced sample, we will not use any inducer. So therefore, there will be no overexpression of our protein of interest. So the protein of our interest, that band specifically will be present in the induced sample only. And if this band is thick, we can understand that yes, our protein is expressed in higher quantity, and this band will not be present in the uninduced sample. So by analyzing this SDS page, we can determine whether our protein is actually overexpressed or not. So that is the correct answer here, B. Next question. Yes, so the protein is 90 amino acids long. So 90 into 110 Dalton will be its molecular weight, which is approximately 9.9 kilodalton. Correct? Next question. Yes. So the correct answer is tyrosine and tryptophan, which are responsible for the absorption of the protein at 280 nanometer. So this is a sum where you have to determine the concentration of the protein, the absorbance, and the molar extinction coefficient is given. So how to determine that? By using the Lambert-Beer law, which is absorbance equals to molar extinction coefficient into concentration. So by dividing absorbance with molar extinction coefficient, we can determine the concentration. And here the correct answer is 0.79 millimolar. So I think you have got this answer, right? When you have divided 9 by 11460, you have got this answer, but that answer you have got in molar, because here you can see that the unit for extinction coefficient is mole inverse centimeter inverse, right? So therefore and the length of the cuvette is 1 centimeter. So I'm just showing you the units here once. So the absorbent does not have any unit. The extinction coefficient has this unit mole inverse or centimeter inverse, and the path length is 1 centimeter, which is constant. So centimeter and centimeter inverse will cancel out. So the denominator has this unit mole inverse. So of course when we divide it, it will come as 0.00079 mole. So that will actually be equal to 0.00079 into 10 to the power into 10 to the power 3 millimoles, which will be equal to 0.79 millimolar. So that is why this is the correct answer, not this one. This will be correct if the unit is molar, but this has the correct unit. So the B is the correct answer. Yes, the B is the correct answer. So is it clear, the unit? Okay, all right. So next question. For salting out of the protein, the salt which is commonly used is ammonium sulfate. That is the correct answer. Next question. Right, to determine the size of the protein, the techniques which can be used can be size exclusion chromatography, SDS page, analytical centrifuge, any of the above. So that is the correct answer, D. Right, D is the correct answer. Okay, now this question 15, here the sequence of the amino acid sequence of this protein has been given. Now you have to state these statements like the molecular weight, the pi, extinction coefficient, solubility. Now, how do you determine this molecular weight, pi, extinction coefficient, these values? So, the tool that should be used is expassy-prot-param. So, if you have access to your computer, please open expassy-prot-param in Google, and there you will be asked to give the sequence. So I will just show you, because I know that this protein sequence is not it's not possible for you to get hand to it. So what I will do is that I'll go to the internet, and I'll share my screen here. So this is actually the expassy-prot-param tool, and in this box we have to paste the amino acid sequence. So this amino acid sequence I'm trying to get. Okay, so this is the amino acid sequence. Okay, I'll copy it and I will paste it here in this prot-param tool, and I will compute the parameters. Now this prot-param will give me the following information. that is what is the number of amino acids, what is the molecular weight. So you can see that the molecular weight is approximately 10,547, so approximately daltons, so which is 10.5 kilodalton. The theoretical pi is 9.99, so approximately 10. We can also get this information of extinction coefficient, which is here 22,460, which is the extinction coefficient, and these other information are also available from this XPASI ProtParam tool. So now the questions that were given is first option was molecular weight is 10.5 kilodalton, which is correct, which we have seen here. Molecular weight is 10.5 kilodalton. The theoretical pi is 10. This is also true. Now the other point was this extinction coefficient 22.46 millimole inverse centimeter inverse. So the extinction coefficient here was 22,460 in mole inverse centimeter inverse. So of course in millimole inverse centimeter inverse it is 22.46. But this particular, so this option, these options A, B and C, they are correct. Now the option D, it states about maximum solubility in a buffer of pH 10. This information is not available, and this information cannot be actually stated. And this information is it's not correct that it's not stated, but it is given that the theoretical pi is of the sample is 10, of the protein is 10. So if the theoretical pi is 10, then the protein will be least soluble in this pH 10 buffer. So that is why this option is not correct, that it is have maximum solubility. It is not correct, because its pi is also 10. So in pH of 10 it will be least soluble. If you have seen this pH with respect to solubility graph. So therefore we can say that this option A, B and C, they will be correct options. So is this okay with all of you, this question, this particular question? Okay. All right. So the correct answer is A, B and C. Now I'm going to the next question. Now it talks about the protein in the previous question. So the pi of the previous protein was 10. So now it is given in a buffer of pH 8. Now how can the protein be purified? Which method should be used? So therefore if the pH is here, the pH is less than the pi. So what will be the charge of the protein, and based on that which chromatographic system should we use? So since pH is less than the pi of the protein will be positively charged, and if the protein is positively charged, then the cation exchange chromatography should be used. In the case of a cation exchange chromatography, what will be the matrix charge? What will be the charge of the matrix? So in case of a cation exchange chromatography, as we had discussed previously, I'm just repeating it once more, that the matrix should be negatively charged in case of a cation exchange chromatography. The matrix is, for example, this carboxymethyl matrix, carboxymethyl cellulose. So the matrix will be negatively charged, so it will be acting as a cation exchanger. So the correct answer is cation exchange here. Now size exclusion chromatography can also be used, and in that case the protein is not purified based on its charge, but it is purified based on its um, size and shape. And affinity chromatography can be used if the protein has 6x histidine tag. So if the protein does not have any tag, so if the protein is untagged, then affinity chromatography cannot be used. But if the protein has a tag, then affinity chromatography can be used. If the protein if you use a buffer system where the protein is either positively or negatively charged, then we can use ion exchange chromatography. And to separate the protein from proteins of different molecular weight, we can also use size exclusion chromatography. Next question. Alright, so the technique which is best used for estimating overall secondary structure of a protein, that means the percentage of secondary structure, should be circular dichroism, from which we can determine the percentage of alpha helix, percentage of beta sheet of a protein. Next question. Okay, the three-dimensional structure of the protein can be determined by which of the following methods? The correct answer is A and B, by NMR spectroscopy and X-ray crystallography. We can also use cryo-electron microscopy, which is also a very good technique for large complexes of proteins. Next question. Okay, particle size and sample homogeneity can be detected by or can be determined by which of the following techniques? Dynamic light scattering, analytical ultracentrifuge, as well as size exclusion chromatography. All three of them can be used for detection of this particle size and sample homogeneity. In dynamic light scattering also, we can determine the diffusion coefficient or the particle size of the sample. In analytical ultracentrifuge also, we can determine the sedimentation coefficient along with the mass of the protein. In size exclusion chromatography also, we can determine the oligomeric state as well as the size of or the molecular weight of the protein sample. And last question. So the protein does not have any affinity tag. Now, what is the best sequence of chromatographic techniques that should be used to purify the protein? and this I had not mentioned before, but when we are using multiple chromatographic techniques or multiple chromatographic steps, we should remember that the protein yield will also become smaller and smaller with each purification step, because in each purification step there will be some loss of the protein. So suppose in the beginning you have done a purification step where you have got 6 mg per ml protein, but in the next purification step the yield of the protein, the concentration of the protein will be reduced. You will get less amount of protein because there will be some loss of the protein. So the more number of chromatographic steps we use, we will get pure protein but in less quantity. So the best purification sequence will be salting out, ion exchange, and followed by SEC. So the D is the correct answer. So what we will do in salting out, the protein will be salted out or precipitated from other proteins by using salt, usually ammonium sulfate or guanidinium hydrochloride we can use, then followed by ion exchange. So the protein is taken in a buffer when it is either positively charged or negatively charged, followed by size exclusion chromatography. So size exclusion chromatography is usually or most commonly the final purification step, because in case of size exclusion chromatography it gives higher yield of the, or sorry, not higher yield, the higher purity of protein is actually we actually get higher purity of protein from size exclusion chromatography. So that is the final step where the proteins are separated based on their size and shape. So this is the ideal sequence of protein purification for a protein which does not have any affinity tag. So because it does not have any affinity tag, nickel column cannot be used, and this option B is not correct because SEC should not be used in the beginning and salting out should not be used in the final step, as a final step. And this is actually how an automated purification system looks like. So here you can see this is actually the protein purification column. So this can be an affinity column, this can be an ion exchange column. Usually size exclusion columns are bigger in length, and size exclusion columns can be of different types also, analytical grade or preparative grade. Now there are these pumps and all these pumps you can see here which actually direct the flow of the protein sample. So here in these bottles you prepare the buffers, you keep the buffers in these bottles, and these buffers are so there are these wires you can see. These wires will be taking this buffers through this protein. So this actually here you can see this is an affinity column. This is an affinity column because you can see this is blue in color. So usually the blue color comes from the nickel. So when since the affinity column contains this nickel matrix. So and then this protein is finally, so first it is being purified by this affinity column, then it is being taken to in this size exclusion column, or it can be ion exchange column like that. And this is called a fraction collector. So where the protein, the elution protein, when it is eluting out, it is collected in fractions. So this fraction collector actually rotates and it keeps on collecting a particular volume of the fraction of this elution, the eluent. Right, so this is an automatic protein purification system, and there will be an UV cell here which will act, and there in this computer there you can see the chromatogram as the protein is being purified in real time. So from that you can see where the peak is coming from the UV absorbance. So this is an ACTA purification system by this company which is called GE. So not all purification systems are automated, but this is an automated purification system where you can purify your protein, and then you can use multiple columns together to purify the protein and finally get a highly pure protein. So that's all for today. Thank you for joining. If you have any questions, please let me know. If you have any doubts, please let me know. And this lecture, along with all the other previous lectures, the 6 weeks of lectures, can be found in this YouTube channel. This is the link, and if you have any questions, any doubts, queries, you can mail me in this email ID. And thank you for joining. Please let me know if you have any doubts. Thank you so much. We will meet again in the next week, Tuesday, same time, 6 to 8 p.m. Thank you.